Wagwan, my GCSE science students, welcome back to another revision video. This time it is for chapter 2, cell division. Okay, if you're AQA, this is the video that you should be watching for chapter 2. Okay, now I'm not trying to do this too quickly. All right, so give you a chance to ensure that you have your flashcards ready. Okay, it is worth the time to watch. All right, really good for your revision. All right, let's go. So the topics that we're going to look at for chapter 2 is cell division, growth and differentiation, stem cells, and stem cell dilemmas. Okay, all right. So here goes. So one of the first thing that you need to understand before you can do or revise chapter 2 effectively is that ensure that you know all of these words. So right here you have a cell, okay, and in that cell you have some thread-like structures. These are called chromosomes. In humans we normally have 23 pairs, 46 in all. Okay, now if you take out a chromosome and unwind that, okay, the chromosome will look like this. So notice now it unwinds into your DNA structure, and a section of that DNA is referred to as a gene, which codes for a particular characteristic. Okay, now you also need to put these in order and be able to put these from smallest to largest. So here goes. Your smallest one would be your gene, and by definition the gene is a section of DNA that codes for a specific protein. Okay, everything in our body is a protein, whether that may be your eye color, your hair color, everything about you is a protein. So all of that information is on your DNA. Next up in size would be your chromosome, and your chromosome is a molecule of DNA consisting of a group of thousands of genes. Okay, and as I said, in humans we normally have 23 pairs, followed by your genome, and your genome is the collection of all the chromosomes that define an individual. Okay, good. Followed up by the nucleus, which is the organelle that contains the DNA and that controls all the activity of the cell. Some textbook would say that it is the brain of the cell. All right, and the cell by definition is the smallest functional unit of an organism. Okay, good. Now why is it that we need to make a copy of a cell? So if you think about it, when you were born we all started as one single cell. Okay, that cell is called a zygote. Obviously today you're not one cell, so that one cell became two, and that two cell then became 4 cells. Okay, and those 4 cells became 8, and so forth until you eventually form a ball of cells, and then so forth and so forth. That ball of cell, by the way, is referred to as an embryo. All right, we're going to be looking at that. But why do we need to copy cells? Now the reason we need to copy cells obviously is for growth. As I mentioned before, we all started out as one cell, all right, referred to as a zygote, and now we are millions, trillions of cells actually. Good. We also need to repair cells. So if you think about it, you probably had damage something, or everyday cells are dying. We need to repair your cells. And finally we need this for reproduction. Okay, good. So that's the reason why cells need to be copied. Now a cell goes through the following life cycle, and we refer to that life cycle as the cell cycle. So in stage 1, as you can see, the majority of the life of the cell is spent here in stage 1, all right, where the cell is replicating. So lots of things happen here. The cell is growing, it's increasing in size, so increase in size. Okay, good. Increase in size, and when it gets towards the end right here, it begins to replicate. The word replicate is coming from the word replicate means to make a copy of itself. Okay, so all the organelles rather inside that cell, all right, you would have your chromosomes, you'd also have your mitochondria, you have your ribosomes. All of those are copied. Okay, copies are made from that. Okay, all right, then on to stage 2. Now in stage 2 you have something which is called mitosis. All right, and what's happening in mitosis is that lots of things are happening. You can use this to remember it. PMAT. Okay, PMAT. So you have prophase. All right, you don't have to know the name of the phases when you're revising. Okay, metaphase, anaphase, and telophase. Okay, good, telophase. All right, all of that is happening. So what's happening in there? We're going to look at that mitosis stage in a bit. Right, the contents of the cells are rearranged. The chromosomes are pulled apart at the end of each cell. Okay, and we're going to be looking at that stage in a bit. And then towards the end of the cell cycle now, which is stage 3, you now have the division of that cell, which is referred to as divided into 2 cells. So the cytoplasm breaks in a stage called cytokinesis, and the cell then divides into 2 daughter cells. Okay, to identical daughter cell. That is a key word there. Okay, good. So here goes. Now, what we're going to be talking about here is mitosis. So in the first part, you have your parent cell. So if you notice here, you have 4 sets of chromosomes there. Okay, good. You can see the different colors. Now that's your parent cell. Okay, so pay attention to the chromosomes there. Then what will happen now? The chromosomes begin to make copy of themselves. So if you notice here, the pink one there makes a copy of itself. The smaller one makes a copy. They all make a copy of themselves. So you can see what's happening there, and they're attached there by something called a centromere. But that's not important now. So right here, what's happening there? That's called interphase. That's happening in stage 1 of the cell cycle. Okay, good. So after it makes a copy of itself, then they're now lining up in the center. So you can see here, right, the chromosomes begin to line up in the center. Okay, so you'd have interphase, then you have prophase. All right, makes a copy of themselves, interphase and the prophase. Good. Then they're lined up in the center here, which is metaphase, right in the middle. So you can use the word meta to remember that is happening in the middle. So the chromosomes line up in the middle, and this is controlled by something called spindle fibers. Okay, so you can add that, especially if this is a 6 marker. This is controlled by spindle fibers. So after they line up in the middle, they've started to now being pulled apart. All right, good. And this is referred to as anaphase. Okay, so anaphase, just to remember that they've been pulled apart or they've been pulled away. Okay, so you can see the anaphase here where they're now being pulled away. After that, the cell begins to divide now into 2 daughter cells. All right, so what's happening here? The cytoplasm is splitting. That is referred to as cytokinesis. Okay, where the cytoplasm is split. All right, good. You can see their cytoplasm is split there, but you also, right before cytokinesis, there is a phase called telophase as well. Okay, good. And right now you now have your 2 daughter cells. Okay, so to remember everything, you can use this acronym to remember everything. You can use IPMAT. Okay, IPMAT to remember everything. So you have interphase, okay, prophase, metaphase, anaphase, and telophase. Okay, good. Telophase. All right, excuse my poor writing. So you can see exactly what's happening there. All right, so just remember now, interphase is not a part of mitosis. So mitosis is from prophase going down. All right, so make sure that you're able to tell what's happening. If you notice at the end, you end up with 2 daughter cells which are identical. Look at the chromosomes here. So we started out with how many chromosomes? 4. My daughter cells also have 4 chromosomes here. All right, so 4 chromosomes here, 4 there, and notice that they are the same, identical. Do not confuse mitosis with another type of cell division, which is meiosis. Okay, now we're now going to look at something called stem cells. So remember what we said earlier. We all started with one cell, and that one cell is called a zygote. That one cell then becomes two cells, and eventually, after several steps of mitosis, okay, you're now going to have a ball of cells. These ball of cells are referred to as the embryo, but these cells are special. These cells are called stem cells, okay? And stem cells means that they're undifferentiated, they're not specialized, but these stem cells can become any other cell inside the body, okay? They can turn into any specialized cell. So if you have a look here, your stem cell can become a fat cell, a nervous, a neuron, a red blood cell, a pancreatic cell, a cardiac cell, okay? And eventually they differentiate. So this process is called differentiation. Okay, so differentiation is the process by which your stem cell becomes specialized. Okay, your stem cells become specialized. All right, now there are two types of stem cells in animals. You have your embryonic stem cells. As the name suggests, they are formed during the early stage of the embryo. They can differentiate into most type of cells. And you also have your adult stem cells, which is taken from a bone marrow. Okay, these cells can differentiate into some type of cells, for example, blood cells. Okay, now it's important that you realize that stem cells is not only found in animals. Stem cells is also found in plant cells, and they're taken from a special region which is called your meristematic region, your meristem. So meristems is normally found at the tips. Okay, so those are your meristematic region. And these cells, they can differentiate into any type of plant cell throughout the life of that plant. Okay, and they're used to produce clone. Okay, so this is why if you cut certain areas and you put them, it can make a clone of that plant. So I'm just showing you the meristematic region. Okay, and that region contains your that region contains your stem cell, which is found in plants, and it's normally found in the roots and the shoots. Okay, now when plants grow and they get taller, it is when these cells in the meristem, they elongate. Okay, following cell division, and this is why plants get bigger. All right, so it's not because of cell division. So we get bigger because of cell division, because of mitosis. Okay, however, in a plant, they get bigger because your meristems are elongated. Okay, good. So here are the key points. So many plants can continue growing throughout their lifetime, whereas we stop. All right, most plant growth occurred by cell elongation, not cell division, and growth in plants only occur in certain tissues, your meristematic tissues. Okay, good. And many cells, plant cells, they retain the ability to differentiate into any cell type. All right, good. This is why cuttings can be used to create new plants. That's why we measure. Now, plant cells. Now, why is it being able to clone large number of plant cells useful? This is super important in the agricultural industry, in the horticultural industry. All right, you can produce a large number of plants to sell. Also, in the agricultural industry, you can produce large number of identical crops. So let's say there's a crop which is resistant to certain type of disease. You can use plant stem cells for that. Also, I would add as well, what about plants which are extinct? If you have plants that are extinct and you have their meristematic cells, you can then save those plants by doing that. Okay? Now, because of stem cells, you're now able to carry out something which is called therapeutic cloning. All right, so you need to be familiar with the term therapeutic cloning. Now, what happens in therapeutic cloning is that, let's say, for example, you have someone with diabetes. Okay, what you then do, you take that cell, a skin cell perhaps from that patient, and you also will have some sort of egg donor from someone with XY chromosomes, a female, okay, biological female, okay, good. You then combine them, all right, then use combine them, and then you grow them into an embryo. All right, so now you have a ball of cells here, and remember that these embryo can now be differentiated into any type of cell under the right condition. You could then get the cells to differentiate into pancreatic cells, which can produce insulin, and then you can then take that and insert that back into the person with diabetes, okay? And then you could then use that as treatment, okay? And this is what we call therapeutic cloning or stem cell research. A very, very powerful thing, all right? Now, why is it used from the same patient? So that it doesn't or reduces the, well, actually, it would not be rejected by the patient's body. Simple, because remember, you'd have taken the cell from that patient. All right. However, there's certain potential risk. Okay. Could you be transferring infection? You have to think about the ethics. Is it right? Is it wrong? And of course, you're going to have certain religious objections. There's certain groups of religion that will think that you're interfering with God's work or Allah's work or anything like that. Okay. Now, stem cell, a very, very powerful stem cell can be used to treat lots of human diseases, okay? Now, you collect the stem cells from the early embryo, as we mentioned before, and before they have started differentiating. And because of this, they can produce any type of cell, any type of tissue, any type of organ. And you can use it to treat so many diseases, okay? You could use it to treat diseases such as diabetes. You could use it to treat diseases such as heart diseases, any eye defects, lots of things that you could use it for, okay? Parkinson's disease, lots of different things, damaged bone, all right? So the stem cells are collected from the embryo, and then the embryo becomes discarded. So that's something that you need to be thinking about. You discard the embryo, okay? So discard embryo. All right, good. The other type of stem cell, we also spoke about the adult marrow one. The operation may be simple, but it's painful. All right, good. Again, it can differentiate into not all the different types of cells, but only some type of cells. Okay, good. Now you need to know the advantages and disadvantages of both of these techniques, whether you're using stem cell from bone marrow or embryonic stem cells. And let's look at that. All right. So in terms of pros and cons for your embryonic stem cell, you can use to treat a wide variety of diseases. All right. You're going to have lots of stem cells available. All right. It is better than wasting the embryonic stem cell, okay? Because sometimes they discard the embryo from IVF, okay? So it's better to use the embryonic stem cells from IVF, all right? And it's also painless. The cons of it is that you're going to damage the embryo after using it, okay? So there are certain ethical questions there, all right? It's a fairly new technique. It may not work all the times. We don't know the long-term effect. All right, as I said before, it is fairly new, and you cannot ask the embryo, because there's some religion or some people would argue that is it alive? We don't know if an embryo is alive. So that's an ethical question. Some people say the embryo is alive. Some people said it isn't, okay? We don't know when life quite started. That's something that's debatable, all right? However, with the adult bone marrow stem cells, there are no real ethical issue in terms of you can't get permission to collect the adult bone marrow stem cell. The recovery time is quite quick. It's fairly safe, and it has been tried. The cons of it is that it only produces a few types of cells, so you can't treat all the problems. It could be painful to the person who is donating it, and also there's a risk of infection, okay? Make sure that you know all of the pros and cons, and that summarizes chapter 2. I'm out.